Yes, it's Uncle Jegs. Last time we looked at antibiotics and painkillers, and also how antibiotic resistant bacteria have made a comeback. In this video, we're going to be looking at drug discovery, but also drug development.
You know the one. You buy one, you get one free. I said you buy one, you get one free. Right now.
Well then, let's chop some science. Here are the retrieval questions for this lesson. You know what to do. We saw from the last lesson that painkillers treat the symptoms of an illness. They don't actually kill the pathogen. Food poisoning is caused by salmonella, which is a bacterium. Osmosis is the movement of water from a low concentration solution to a high concentration solution. Bare students always get osmosis wrong, and if you were one of them, clickety-click the card to teleport to that lesson. The top image here shows a willow tree. We also have a piece of text telling us that willows produce a precursor to aspirin. The bottom picture shows a beaver having a random snack. The challenge question is asking you to state why chewing the glands from the beaver's tail can help to relieve pain. Seems a bit random, but you should be able to work that out. Beaver's tail was actually one of the few ingredients that witches used to use in their spells and potions.
Yeah, boy. That's it.
The more challenge question builds upon the previous question and asks you for 2 marks why the populations of beavers decreased in some regions. And finally, the mega challenge question asks you to suggest 2 methods that do not impact the population of beavers. You have to have the previous question correct to get this one. Pause and have a go. Beavers eat the willow tree bark. The painkiller gets stored in their gland after they've ate it. The beavers are being killed before they reproduce. This will reduce the population size. And finally, you could either use the willow tree directly, or you could extract the painkiller from the beaver without killing it.
I risk my life.
In the specification, you need to know the origins of 3 drugs: digitalis, aspirin, and penicillin. Digitalis is extracted from a plant called foxglove. Digitalis strengthens your heartbeat, so it's given to patients that have issues with their heart. Aspirin, as we saw, is extracted from a plant, the willow tree. The technical function of aspirin is that it's an anti-inflammatory. When your body cells or tissues are irritated or damaged, your body produces an inflammatory response. This causes swelling. So anti-inflammatory drugs reduce pain by reducing the inflammation around the damaged area. This is different from other drugs like opioids that actually block the pain signal going to your brain. Finally, as we saw in the previous video, penicillin comes from a fungus, but you can also call it a mold. And it kills bacteria. Before we look at how modern day drugs are developed, let's recap what Alexander Fleming actually did.
Oh wow.
Oh wow.
I deserve some accolade.
Yeah, Prof, you do.
Normally, bacteria grow all around your petri dish. Fleming noticed a lack of bacteria growing near the mold, and that was this mold here, the white stuff at the top. So he realized that the mold was producing an antibiotic that was killing the pathogen. Although scientists make most of the drugs in the lab now, they still start from raw materials from plants, microbes, and some animals. For example, here is the last part of a reaction that creates aspirin. This molecule over here is aspirin.
Wool up, just wool up. What's a business, man?
You obviously don't need to know this chemical reaction, but you do need to be able to state that most drugs are now made synthetically, or they're made in the lab. There are 4 properties that make a drug an excellent drug.
Cocaine is a hell of a drug.
No, Mr. James. Kids, do not do drugs. On the right, I have the meaning of these 4 properties. Take a moment to match up the properties with their corresponding sentences. An effective drug prevents or cures the disease. A safe drug isn't very toxic and doesn't have unacceptable side effects. A stable drug can be used and stored under normal conditions. And a drug that you can excrete means that it can reach its target, do its job, and then be cleared out of your body through your wee or poo.
Wow! Let me go home.
Let's play a little thought experiment.
Imagination.
Let's say that you notice a bacteria growing near some cancer cells that you have on a petri dish, and you notice them killing the cancer cells just the same way Alexander Fleming noticed the mold killing the bacteria. The microbe that you're looking at produces thousands of compounds, but you think there's only one compound that's killing them. These are the resources that you have. You have lab rats, human cells and tissues, healthy volunteers, and cancer patients. There are 2 testing stages that you need to do. You have your preclinical testing, and then you have your clinical testing. Preclinical testing occurs before the drug is tested on humans. This involves testing the drug on cells, tissues, and live animals. Let's see the impact of preclinical trials. When the drug was discovered, you have a possibility of up to 10,000 compounds that could be your active drugs. But during the preclinical trials, we reduced the possible compounds that it could be to roughly 20 compounds. The clinical trials are when the doctors and scientists are ready to start testing the drugs on healthy volunteers and patients. This is the full process of the clinical trials up to when the drug is licensed. Let's take each phase one by one. The first phase of the clinical trial takes roughly 1.5 years and reduces the possible compounds to around 10. Here are some challenge questions. You know what to do. The first question was slightly a trick question. The first stage is the preclinical trials, what we saw in the last slide. At the first phase, you should use low doses because the compound could be toxic when you inject it or give it to humans. This next question requires a little bit of thought. If the drug is dangerous, you wouldn't want to expose your patients who are already weak to the drug. So at this stage, you use healthy volunteers because the side effects of the drugs could damage or kill your patients. For your exam, you need to be able to summarize phase 1 as testing for toxicity. Now on to phase 2, which takes roughly 2.5 years and narrows the possible compounds to a possibility of 5. Also, I have this image on the top that will provide some useful information for the following challenge questions. The first question is asking you what a placebo is. The image will assist you with this. Have a look carefully at that. The second image will also help you answer the second question, which is what is a double-blind trial. And finally, this stage is when you start using actual patients, which will help you understand what this stage is testing for. Try and answer all the questions. A placebo is a medicine that doesn't have the active drug. You can also call it a fake drug. A double-blind trial is one where both the doctor and the patients do not know if the patient is getting the drug or the placebo. Since we're using patients with the illness, we are seeing if the drug actually works. The key word for this that you should use in your exams is efficacy. So phase 1 was testing for toxicity, phase 2 is testing for efficacy. Now we're on the last phase of the drug trials, and the drug now has been narrowed to roughly 2 drugs. These are the last set of challenge questions. You know what to do. In the third phase, we want to use more patients because it could have a negative effect on some patients but not on other patients. At this stage, we increase the dose so that we can see what the best dosage of the drug is. And finally, other companies should repeat the results to avoid biases from the drug company. This is very important because some of these drug companies move bare snaky. Make sure you memorize the function of each phase. Phase 1 tests for toxicity, phase 2 tests for efficacy, and phase 3 tests for dosage. And there we go, lesson done. We looked at the origin of 3 different drugs, and we also looked at preclinical trials and clinical trials. I hope you found all of that useful. Check the timestamp below if you need to jump to a particular section, and I'll catch you guys in a bit.